Hello everyone, hope you all are doing well. So today we are going to discuss the next answer copy of our Aim for Agriculture series. So as you all know that we are having Aim for Agriculture series in which we are having daily answer writing practice and weekly full tests. And we have discussed all the answer writing practices, daily answer writing practice of Aim for Agriculture series. And today we are going to discuss the last test copy, that is test number 14, full test number 14. So there were 52 daily answer writing practice copies and 14 full test copies. So we have discussed all the 52 daily answer writing copies and 13 test copy have been already discussed. So today we will be discussing the last full test copy. And all these copies, the PDFs of all these copy have been uploaded in the Telegram group. So you can visit the Telegram group and get these PDFs, because many of you were messaging me to get access to the PDF. So those have been uploaded to the Telegram group, so you can visit and get these PDFs. So let's start. But before starting, I must let you know that as there will be a lot of questions to be discussed, I will be discussing the important points only, the structure of the answer, the important how to structure your answer, that will be our approach. And if you want to read the answers in detail, you can simply pause the video and read the answers. Simply pause the video and read the answers wherever you want to. So that can be helpful to you. So let's start. Or either you can directly download the PDF from Telegram and read those PDFs if you want to. So let's start. So the first question is, give a brief description of plasma membrane structure with level diagram and its function. So it is a well-labeled diagram of plasma membrane. We all know what plasma membrane is. It is a semi-permeable membrane. Now, we will discuss about its structure. Here we will see a phospholipid bilayer. There are proteins here. Cholesterol could be seen. Carbohydrates can be seen. Now there are various functions of plasma membrane, such as, first of all, we all know that it is selectively permeable. It helps in cell communication. It helps in cellular homeostasis. So these are some important functions. So very well written answer. Now let's move to the second question. The second question is, explain inbreeding depression and its effects in crops, and also explain degrees of inbreeding depression. So what inbreeding depression is? It refers to the reduced biological fitness in a population due to inbreeding, that is breeding into the same generations, or you can say in same ancestral generations again and next, or you can see inbreeding within the same genetical or similar genetics, that leads to inbreeding. Okay, so that leads to inbreeding depression. Now it is generally prominent in cross-pollinated species. Now causes have been mentioned, that is increased homozygosity and loss of heterozygosity. So now what are the effects of inbreeding depression? There will be reduced growth and development. There will be lower fertility and seed production. Inbred plants tend to be more vulnerable to pest and diseases, drought, heat stress, various kind of stress. One of the most direct effects is the significant decrease in crop yields. The crop yields will be decreased. Now there are various degrees of inbreeding depression in self-pollinated crops. There are partially self-pollinated crops, cross-pollinated crops. So you can see that in self-pollinated crops you will find that they are generally less tolerant. They are generally more tolerant to inbreeding depression, whereas partially cross-pollinated crop we will be seeing moderate level of inbreeding depression, but and cross-pollinated crops we will see severe inbreeding depressions present. So these are the various levels of inbreeding depression, that is no inbreeding depression, moderate level, and then severe level. The next question is, what do you understand by passport data of genetic resources? And we have to write its importance in germ plant collection and conservation. So as the name suggests, passport data, that is the various information of the organisms, or you can say various essential information collected about the genetic resources have been mentioned here. Okay, such as origin, location, environmental conditions, character, everything has been well mentioned. Examples, for example, accession number, species information, various kind of things have been mentioned. So what are its importance? It helps you to precisely identify the origin and diversity of the germ plant by documenting the geographical and environmental origin. It aids in capturing wide range of genetics that can be done If a variety from Rajasthan shows salt tolerance, it allows, it is the example of this documenting only. It ensures the same genetic resources isn't collected multiple times because we have information and similar things will be prohibited. Okay. What are its importance? It ensures the germ plum is conserved. So germ plum conservation is one of the roles. Okay. It helps in locating the correct genetic resources. It helps in various future research. Okay, so these are some of the importance. Let's move to the next question. The next question is origin and domestication of rice. So generally, in rice is to be said to be originated from Asia and particularly from India and Japan. Okay, and the oldest evidence of rice is Yangtze River Valley of China. It is the oldest cultivated evidence of rice. Okay, so you can simply pause and read it. Then it has been domesticated into various places. Okay, so even this is a fair written answer. You can simply pause and read it. It has been seen in Indus Valley also. I think rice was called virhi in Indus Valley. I'm not sure about, but it was called virhi or something similar to it. Godumi, wheat was called Godumi. Godumi was called to wheat. And this was a UPC PYQ in prelims also, that what are virhi and Godumi. So these are generally cereals of Indus grain. Now let's move to the next question. The next question is explain the method of linkage detection in recombinant breeding program. So how can we detect the linkage in recombinant breeding program? We can draw linkage maps. Okay, linkage mapping can be done. Marker assisted selection can be done. UTL mapping can be done. Linkage disequilibrium analysis can be done. Recombinant frequency analysis can be done. Bulk segregant analysis can be done. Genome wide association studies can be done. These are the things. You can simply pause and read it. Now let's move to the next question. The next question is write in detail the anatomy and cytology of stomata and also explain the factors affecting stomatal movement. So you can see anatomy, the various types, various structure of stomata have been well labeled. The diagram of stomata well labeled, so it is very good. And in anatomy, you will see the guard cells are present, and subsidiary cells are present. Everything has its own functions. Stomatal core is present, epidermal cells is present. Cytology, that is cells present in stomata. So you will find cell wall present, you will find chloroplast present, you will find vacuoles present. So these are the structure of cells. Ion channels are present. Now, what are the factors affecting stomatal movement? Obviously, light, CO2, water availability, temperature, hormonal regulations. So these are the factors which affect stomatal movement. You all know humidity is another factor. Circadian, uh, circadian rhythm is another method. You can simply pause and read it. Now let's move to the next question. The next question is describe systematic acquired resistance, that is SAR, and source of disease resistance with suitable examples, and write the advantages of breeding resistance in plant. So what is SAR? It is a plant defense mechanism that provides broad spectrum long lasting protection after an initial localized infection. Suppose the this is the plant and there is a initial small infections. So what happens when these infections happen? The next time the plant develops kind of SAR, that is resistance is developed, and next time this infection is prohibited and the bigger cause of this infection is prohibited. Okay, and what are its mechanism? There could be salicylic acid accumulation, you will see pathogenesis related proteins, you will see systemic defense, we will see priming mechanisms, well written points. Now what are the sources of disease resistance? It could be natural resistance, it could be horizontal resistance, it could be vertical resistance, it could be induced resistance. Now, what are the advantages of breeding for disease resistance? Obviously, it will cut down the use of pesticides. For example, there are certain kinds of their resistance because of the pests, and if you have breeded the plant with any kind of disease resistance or pest resistance, the application of the chemical used for disease control and pest control will be reduced. Resistant varieties maintain yields. You will see good amount of yields of good variety, good resistant varieties. Okay, they promote environmental friendly farming. Obviously, there will be less use of chemicals. So, well points, you can simply pause and read it. Let's move to the next question. The next question is what are the components of genetic variance and how are these components estimated? So genetic variance in a population refers to the variation in phenotypes that can be attributed to genetic differences among individuals. Now, what are the main components of genetic variance? We all know that we have additive genetic variance, we have dominance genetic variance, we have epistatic genetic variance. And if you are not able to understand the basics of these things, because I will not going to explain anything in detail, you can refer the static videos. I have discussed everything in detail in there. Either you refer static videos, or for answer writing practice, you can also refer the answer writing practice series videos. Okay. So in these two videos, I have explained everything in detail. I am just giving you an idea of structure here, how you can write the answer. And the answer copies here I am bringing is up to the mark. The best copies have been discussed to you, so you can learn from these copies. So this is the idea behind this series, this program. Environmental variance is there. Now, how can you estimate? We can do phenotypic variance analysis, we can do genetic correlation studies, we can do linear mixed models are there, twin studies can be done, QTL mapping can be done. So well written answer. Now let's move to the next question. The next question is, what is bud dormancy? Explain its causes and means to break it. So what bud dormancy is? So what happens when the plant bud is inactive? Dormant was, for example, in seed dormancy, the seed is inactive, and bud dormancy, you will find the bud is inactive. So we have to break this dormancy so that it doesn't prevent the growth, and the buds or the anything rising from the buds rise nicely. Okay. And various times this dormancy is seen in the plants due to various environmental conditions, due to stress, they become dormant. So we have to, for breaking the dormancy, uh, yes. So first of all, we have to discuss the causes. Okay, the causes could be environmental factors, it could be hormonal regulation, it could be any kind of stress, it could be developmental signals. Now, how can we break this dormancy? We can do cold estratification, that is, cold treatment can be done. Thermal treatment, that is, heat treatments can be done. Hormonal treatments can be done. Moisture should be made available. By soaking the moisture, the dormancy could be broke. Mechanical scarification is done, that is, through by applying pressure, we will mark a scar so that the dormancy is break, and everything which is stopped due to dormancy is now again regained or starts growing. Now, next question is, enlist the role of auxins in agriculture with suitable examples. So we all know these types of answers. All the plant hormones have been asked several times, and these are important, so you should cover them. We all know that auxins stimulate root function. They have a role in fruit growth. They maintain apical dominance. So they are similar, simple points. You all know. I think every serious agriculture student should write these types of answers. So you can simply pause and read the points if you want to. It helps in cell elongation. It affects plant height structure. It helps in seed germination. So these are some of the basic points when it comes to auxins. They influence flowering. The next question is, write the causal organism nature of damage and management practices for damping off in vegetables. So what damping off is? That is rapid decay of young plants. Okay. Now it's casual organism. It is generally caused by fungi. It is also caused by bacteria, and you can see the names of fungi, the names of bacteria have been mentioned. The crops have been also mentioned in which crops they affect. So very well written points. Now, what are the measure of damage? They can they they can reduce the problem of, or they can increase, not reduce. They can increase the problem of germination. Okay. You can see damping rot before germination. Post-emergence problem can be seen. Seedlings will exhibit extended growth. So these are some of the problems. Now, how can we manage the? We can do soil management. We can do sanitation practices. That is keeping everything hygienic and clean. Seed treatment using fungicides, but or you can use certified seeds. So well written points. You can use other microbes for 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 correcting them or for treating them. The next question is, briefly discuss the integrated pest management in cotton. So what IPM is, that is integrating different types of methods, agricultural, biological, physical methods to physical and chemical methods to control anything. For example, that is what integration is. And these types of questions have been asked several times, and I think you know here only students have discussed cultural control, biological control, mechanical control, chemical control, host plant resistance. So all these are the factors of IPM only. You can simply pause and read the points. Next question is, explain the biological control methods of tomato, fruit borer, and guava teal. So in biological control method, generally we use the living organisms for treating anything. For example, here we have to treat, or we have here have to control the tomato fruit borer and guava beet. So for tomato fruit borer, we can use Trichogarma species. BT can be used, BT species, that is Bacillus thuringiensis. It is also used in cotton BT, cotton we all know. And PV can be used, that is nuclear polyhydrosis virus. Okay, Saro, Cairo, Spira perla can be used. Entomopathogenic nematodes can be used. For guava wilt, again, you can use Trichoderma species, you can use Pseudomonas species, Bacillus species, neem can, all these things can be used. So you can compost tea can be used. So you can simply pause and delete. The next question is explain the biosynthesis of gibberellins and writes its role in commercial usage in agriculture. So you can see how beautifully the biosynthesis of gibberellin have been described using the diagram, and I think you should refer the PDFs because you will be able to give more time and note down these diagrams. That can be very helpful. So you can also pause here and uh, make these diagrams or not, or have a quick look on this diagram. That can be helpful. So here we see terpene pathway, cyclization is there, oxidation is there, modification is there. And I must say here, whenever you make a diagram in your answers like this, try to explain bit as the student done here. Try to explain in a brief only, but try to write something about it that enhances your answer. Now, what are the role of gibberellins? It has role in cell elongation. It helps in breaking dormancy. It promotes flowering, it promotes leaf growth, it inhibits root elongation. So I think you all know it. As a serious agriculture student, you should be knowing it. Now, what are the commercial uses of gibberellins? They help in rapid, uniform, and seed germination in crops, used for enlarged fruits. Okay, applied in induced spotting. So these are also some of the commercial applications. I think some varieties could be mentioned here, or you can say some kind of more specific examples. For example, you can see examples of cucumber have been mentioned. So specificity have been to be introduced. How it is helping in cucumbers. Okay, although it is mentioned that it can produce more male flowers for hybrid seed production for, and it can be seen. It is fair, but still you can write some specific things also. Okay, but it is good. Okay, it is fair. Okay, you can write like this. And the next question is discuss the package of practices of marigold. And I always tell you that always do keep in mind that the package of practices are very important, and every year 2 to 3, at least two package of practices will come. So you should write all the you should keep in mind all the package of 24 MSP crops, important fruits, all important horticultural crops. So all the things which can be asked in previous year, every any crop is going in news. For example, recently you can see mustard, Dhara mustard 11 is in news. So you should prepare it. Millets have been in news due to UN. You should prepare it. So all these things are helpful and they are asked. Okay. So again, similar things we will use for package of practice. The similar structure can climate, soil, varieties. So you can simply pause and read it. And most of the things are similar here. In package of practices to remember things, pick out the differentiators. For example, in irrigation, so in various flowers you will see similar kind of irrigation, fertilizer application is similar. So we have to pick out the differentiators which are different in for example varieties. The varieties will be different for everything. So pick out the differentiators and mark out the common points so that it could make you easier to remember. Now the next question is write the preparation of ready to serve beverages and squashes. So how can we prepare? The ingredients have been mentioned. The preparation is steps are that first of all we will extract the fruit pulp, then we will prepare syrup, then we'll add citric acid, then we'll add preservatives, homogenize and pasteurization needs to be done. So you can then we have to cool and package, storage should be proper. So written points you can simply pause and delete. Again you can see the another question is discuss the cultivation practices of cauliflower. Here you will see the more horticulture crops are asked because this was the full test of paper 2. In paper 2 you will find generally higher number of horticultural crops in package of practices. In paper 1 you will find cereal crops. Okay, so keep these things in mind. So again same things have been used, you can simply pause and read it, same structure have been followed. And I have discussed I think package of practices, techniques of package of practices several times that I think you should now try to understand differentiators uh, and try to develop an idea to how to approach package of practices because here you can fetch good marks. The next question is how food production, including horticultural crops, changed in India during the last one decade. Write down its impact on food and nutritional security. So we see that there is surge in production, rise in high value crops, adoption of advanced technologies have been done, government initiatives have been there, such as National Mission for Horticulture. You can see PM Kisan scheme is there, Horticulture Infrastructure Development Program is there, Mission for Infrastructure Development for Horticulture. So you can simply pause and read it. Now, what are its impact? Obviously, food availability will increase, hunger will reduce. These products will give us stabilized market prices. Employment will be there. Even horticultural crops are good in vitamins, macronutrients, so they will provide biodiversity in our dietary needs. So you can simply pause and read it. Again, here it is mentioned in detail how it has changed on nutritional security. Obviously, our health will be more enhanced now because they are rich in macronutrients, various proteins, various minerals, various fibers. So that is good only. So you can simply pause and read it. Now the next and last question of this series, that is write a note on malnutrition among women and children. So we all know that obviously in India a lot of women and children are suffering from various kind of micronutrient deficiencies, either it be anemia, either it be stunted growth. So all these things have been mentioned. You can simply pause and read it. Initiatives have been also mentioned, such as National Health Mission, supplementation programs have been mentioned, nutrition educational should be provided, similar for children also. You can simply pause and read it. The resistant being wasting, underweight, lack of vitamins and minerals, obesity have been rising, consequences have been mentioned, initiatives have been mentioned. So that is all for today. I hope you liked this aim for agriculture series, and the next series will be most probably start from November. So anyone who is interested in joining the next series, you can message us on the Telegram or WhatsApp, and you can download the PDF from the Telegram group. All the PDFs have been down uploaded there. So that is all for today. If you like it, please press the like button. Please do share and please do subscribe. It takes a lot of hard work to make such things, make such videos for you on regular basis. And if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly and bring more such courses for you regularly. So that is all for today. Have a nice day. Thank you.